Okay, so this video is about DNA sequencing. So first, you're gonna start off with some isolated DNA. So I'm drawing here. But you only want a segment of the DNA. So we're gonna take this segment, like this, and on either side of that, they're gonna be primers. The orange, so that top and that bottom, so like complementary. Okay, so once you have that, you're gonna put them into these 4 tubes, and so you're gonna put the isolated DNA with the primers in the tubes. So you have DNA, more DNA, and then you put the primers. I'll show an arrow. Okay. So another thing you put in the tubes is DNA polymerase. So you put that in all 4 tubes. And in each, well, I guess in all the tubes, you put DATP, you put DGTP, DCTP, and DTTP. Now in each separate tube, like for tube A, we're going to put DDATP, and then C we will put the DDCTP, and the G will put the DDGTP, and the T will put the DDTTP. So once those all the components are in the tubes, you put that into a hot water bath at 95 degrees Celsius so that they can denature. Okay, so once they are in the hot water bath at the 95 degrees Celsius denaturing, what happens is that in each tube, so this is like for every tube, so like for example, if this is the A tube, you'll have the DNA strands, you'll have the primer, let's say the primer is, oh you can't see that, just kidding, A, G, G, T, C, so the bottom will be complementary, so T, C, C, A, G. So the DNA polymerase comes in, we'll have this as our little DNA polymerase. It comes in and it starts building off the primer. So it's going to start building just randomly, like an A and a C and a T and an A and a G. But what happens is when the ATP comes along, it's going to come and it's going to put an A right here. So the A is what stops the DNA polymerase from putting more nucleotides on the strand. So that's why you get the different links in the strand and then gel. So that happens in tube G. So the same thing happens, but so you have your primers, and then T, C, C, A, G. The polymerase comes in, it starts building randomly, but it's going to come to a G, and the polymerase is going to stop. I'm going to draw my polymerase. It's going to stop because of the DDGTP and so on and so forth. So that happens in tubes T and C as well. So once they've been in the hot water bath, you take them and you put them in different wells of the gel. So you have like A, C, T, and G. And if you do it the old time-year method, you put them in a gel and then the gel hangs on from a wall, and they start going down to the negative side, positive, negative. They start going down, all of them go down, I'll put one more on C. So the way you read how you get the DNA sequence is you start from bottom to up. So if it's hanging, you're going to read from bottom up. So the sequence will start like C, and then it's going to go, add one right there to tell, to T, and then to G, and like another C, T, and G, and then it's going to finally get to that A, and there's your sequence.